Good morning, everyone. Thanks for staying so late at this. I know I'm the last day, so there aren't as many people here, but we have a really exciting story to share. And okay, I guess I don't have it here. All right, so my name is Baughn, and I am one of the co-directors of the Genomics Laboratory at the Mayo Clinic. And this work was led by Ala Koleilet, a fantastic fellow of ours at the Mayo Clinic. I have one disclosure to reveal on this slide. So as most of you are aware, multiple myeloma is a plasma cell neoplasm. It is the second most common hematologic malignancy. There were about 35,000 new cases in the US in 2021. This is a malignancy of older adults with a median age of diagnosis of 65 years and remains incurable. What is significant and rather devastating about multiple myeloma is that it is associated with a fairly significant health disparity. So at this point, I want to make the comment that self-report race categories represent a social construct. In the US, health data are collected in these categories, and these data have revealed that multiple myeloma is the most common heme malignancy in individuals who self-report as Black or African American. The age-adjusted incidence of myeloma is 2 to 3-fold higher among African American individuals compared with non-Hispanic white individuals. In addition, African American individuals have a higher mortality outside of equal treatment access opportunities. The reasons for this disparity are very complex and complicated, resulting in factors such as socioeconomic, social determinants of health, structural racism, bias, environmental, behavioral, and biologic factors. What is also significant to note is that Black individuals are underrepresented in research studies, and this most likely contributes to the health disparity. Black individuals are only represented in 5% of clinical trials and 2% of translational research studies. In addition, there is evidence to suggest that there is reduced quality and quantity of sequence data. For example, the underdetection of genomic variants and as well as the inflation of tumor mutation burden, which was also raised yesterday at a talk in association with African Ancestry. So we specifically asked, do clinical diagnostic testing approaches that utilize genomic data display differential performance between African American and non-Hispanic white individuals? As you know, most of the time, the current testing approach for multiple myeloma does involve fluorescence in situ hybridization. So the reason why we perform FISH is because these genomic abnormalities influence disease course, response to therapy, and disease prognosis. So I'm going to talk about 2 interesting observations that we made using these data. So I mentioned several years ago, actually at CGC, our concordance study where we compare the performance of mate pair sequencing to FISH and found that 6 of 70 tumors had a discrepancy between these technologies for the enumeration of chromosome 15 using the D15Z4 probe. So in the next 2 slides, I'm going to show you how this discrepancy looked. So in the first example, by FISH, we see 1 copy of the D15Z4 probe, and by mate pair sequencing with the red box indicating chromosome 15, you see that there are 2 copies of chromosome 15, a normal signal pattern for chromosome 15. In the second example, you see by FISH that we have also 1 copy of the D15Z4 probe, but in this case, we actually have a trisomy 15 result indicated by mate pair sequencing. So we asked, what is the incidence of this discrepancy? So approximately 2017, we had built a large, diverse cohort, including 1,224 individuals with an abnormal plasma cell FISH result. So instead of using self-report race data, we now turned to calculated genomic ancestry. We utilized a biogeographical ancestry prediction using the Geographic Population Structure Origins tool in collaboration with our population geneticist, Aaron Alhike. we are able to estimate individual ancestry in relation to 36 admixture proportions representing 10 African components. Using these data, we find that in our cohort, 27% represent individuals with over 50% African ancestry. We found a monosomy 15 result in this cohort in 49 cases, representing 4% of our total cohort. Next, we asked, what is the nature of this discrepancy? Of these 49 cases, 42 cases had light chain counterstain data, and 3 cases had monosomy 15 only present in the plasma cell compartment, representing a somatic plasma cell-specific event. In 39 cases, we had monosomy 15 present in both the plasma cell and non-plasma cells. Of these 39 cases, 37 had a complete chromosome study, and of 740 total metaphases, 737 had 2 normal chromosome 15s. In addition, metaphase fish demonstrated a failure of the D15Z4 probe to hybridize to 1 chromosome 15 centromeres in a cell with 2 chromosome 15s. So in terms of the evidence that this monosomy 15 fish result is a true false positive, we demonstrate a clear discrepancy between fish and pair sequencing. We find the presence of monosomy 15 in all cells analyzed in the bone marrow, not simply those restricted to the plasma cell compartment. We see the presence of 2 chromosome 15s by chromosome studies, and also the strongest evidence is that metaphase fish demonstrated a failure of the D15Z4 probe to hybridize to 1 chromosome 15 centromere. So we next ask, is this phenotype specific to a certain population within our diverse cohort? And this comes to our second interesting observation, which was actually truly incidental. So when we grouped the cohort into those that had a monosomy 15 result versus those without, we see a very significant enrichment of African ancestry. In those cases that had monosomy 15, we see a median African ancestry of 77.2% in comparison to 2.2% in those without a monosomy 15 fish result. Using a logistic regression model, we showed that individuals with African ancestry have an 8.02-fold increased probability of having a false positive monosomy 15 fish result. So in those individuals with over 50% African ancestry, 12% had a false positive monosomy 15 fish result. In addition, we utilized the 10 different African components in our ancestry analysis and found a lower contribution of the Nile Valley peoples, as well as increased contribution of Northwestern Africa in cases with a monosomy 15 fish result. Because we had previous data showing that we have an enrichment of the TLM14 primary cytogenic abnormality in association with African ancestry, as well as an increase in the germline SNP RS9344, which is associated with TLM14, and this is also enriched in African ancestry. Our hypothesis was that most likely those cases that had a monosomy 15 fish result would also have an increased incidence of a TLM14 translocation, and we were able to demonstrate that as well as a reduction in the overall trisomy hyperdiploidy category. We also performed a logistic regression analysis and showed that African ancestry was significantly associated with monosomy 15 after we adjusted for age, sex, and cytogenetic subtype. We're also interested to know a little bit more about the mechanism for how to explain our findings. We utilized structural variation data from the 1000 Genomes Project. We found that there were 8 deletions identified within the 15p11.1 to q11.1 region. 6 of these deletions were found in individuals of African descent versus 2 to 4 were present in the other 4 populations. The sequences of the 6 deletions did reveal a high identity with the 171 base pair consensus monomer of the AT-rich alpha satellite repeats. In addition, the African alpha satellite deletions were much larger in comparison to the deletions found in the European population. So in conclusion, we report a polymorphic variant of the chromosome 15 centromere, resulting in a false positive monosomy 15 FISH result. The incidence of the true monosomy 15, which I didn't talk about here, is actually fairly low. It's approximately 0.4%. So when one sees something like that, these cases are most likely associated with the high-risk hyperhaploid subtype of multiple myeloma. In the lab, if you see a monosomy 15 result and it's found in all of the cells in the bone marrow, you may be concerned for the possibility of this being a polymorphic variant that we have shown here to be enriched in individuals with African ancestry. We also acknowledge that the incidence of this false positive finding is likely greater since we were not able to evaluate cases that had a true trisomy 15, but actually had a reported normal chromosome 15 finding. Although I did not emphasize the clinical significance of this finding rather than focus more on the technical aspect, if one is utilizing a probe like this, this could have, may actually increase the risk of an inaccurate diagnosis of hyperdiploidy in the context of myeloma. So based on these findings, we feel that it would be important to utilize diverse control samples when validating FISH probes and other diagnostic testing methodologies. So finally, I would like to acknowledge all of the fantastic people in the clinical genomics laboratory, as well as our population geneticist, Erin Alhike, whom we work very closely with, and the individuals in the research laboratory, the clinical collaborators, as well as my funding sources. Thank you very much.